biology in the unit, second unit, cell, the unit of life. So in this second unit, this is the second part of the lesson. So already we have saw about what is cell and what is cell theory, and about the prokaryotic cells we have discussed till now. So the next part of the lesson is about eukaryotes. So what is eukaryotes? What are the organelles that are present in eukaryotes? All those things we are going to see in this video now. So let me start with the eukaryotic cell. So what is eukaryotic cell? As I have already told you, eukaryotic cell means those which have well developed cell organelles are called as eukaryotic cell. So well developed organelles means what? They will have a nucleus to control the cell, and after that they will have a mitochondria to give power, and vacuole is present, the endoplasmic reticulum will be present. So each organelle will be present for specific function. So that is only considered as a well developed cell. So those cells are only called as eukaryotic cell. Now, where will these eukaryotic cells will be present? It will be present in protists, plants, animals and fungi. So protist means the protista category that will be coming under the animal kingdom, and plants, animals and fungi. So in these types of organisms, the eukaryotic cell will be present. And next is well developed cell organelles are called as eukaryotic cell. Now let me tell you about plant cells. In plant cells, the vacuoles, the empty spaces, there will be huge large vacuoles in plant cells, and in plant cells a covering called cell wall will be present. So this is about plant cell. And in animal cell, if you see, means the vacuoles won't be large as plant cell, and the cell wall will be not present. So only the plasma membrane will be present in the animal cell. Cell wall will be present only in the plant cell, whereas if you see animal cell, the plasma membrane only will be present, and very small vacuoles only is there. But in animal cell, you can find a structure called centrioles. So that will be present in animal cell but not in plant cell. So these are some of the distinct features of plant cell and animal cell. So now let me move into the plasma membrane structure. So now we are going to see each and every organelle of this eukaryotic cell, how the structure will be, what it is function. So those things we are going to see now. So plasma membrane, if you see, means it is mainly made up of carbohydrates and proteins and also lipids. So this plasma membrane, it is a bi-lipid layer. So two layers of lipid are there. So that is why it is called as a bilayer. Phospholipid bilayer. Lipid means fats. So two layers of fats are there. So it is called as phospholipid bilayer, and below you can find integral proteins, and these round structures are the phospholipid head, and this is the phospholipid tail. Then cholesterol will be present here and there, and finally glycoproteins are present. So this is the chemical structure of plasma membrane. What is plasma membrane? So plasma membrane means it will be present after the cell wall in case of plant cells, but in animal cells it will be the outer covering of the whole cell, and it is semi-permeable membrane. So what is semi-permeable membrane? It will allow only certain substance to go inside and go outside. It is like a security guard to the whole components of the cell. So all the molecules cannot enter the cell. It will detect what type of component it is, and then only it will let into the cell. So this is the main function of plasma membrane, and we have seen the structure. And it is made up of phosphoglycerols. So it is made up of phosphoglycerides. And 52% of protein and 40% of lipids are present in human erythrocyte plasma membrane. So all these cells have plasma membrane. So human erythrocyte means the red blood cells, the RBC. So RBC only makes the blood red color. So in that human erythrocyte, the plasma membrane will be made up of 52% of protein and 40% of lipids. So this is the composition of human erythrocyte plasma membrane. And the two types of plasma membrane that are present is peripheral protein and integral protein. So these are the two types of proteins that it will be present. So peripheral protein will be present inside the plasma membrane, whereas integral protein will be buried inside the layer. So this is the position of the two types of proteins. And after that, fluid mosaic model. So this fluid mosaic model, it was based on the structure of plasma membrane, that is how the structure is there and what are the components that are present. So based on only the structure of the plasma membrane, this fluid mosaic model was discovered. So in 1972, this model was discovered by Singer and Nicholson. In this fluid mosaic model, what they are trying to tell is, what is the main aim is, how the components move inside and outside the plasma membrane. So I already told you, this plasma membrane is semi-permeable. It will detect all the molecules to move outside and inside. So based on that, how these components are moving inside and outside. So studying mainly about that only is called the fluid mosaic model, and it was put forward in 1972 by Singer and Nicholson. And next is passive and active transport. So passive transport and active transport. So these are the two types of transports in which how the molecules are moving. So the movement of the molecules are divided into two types. One is passive transport and another is active transport. So what is passive transport? Passive transport means for the movement of the elements, they do not need energy. So they naturally have the ability to move. They do not need extra energy for the movement. So such type of transport is called as passive transport. Whereas active transport, if you see, means it will need energy for the movement of the elements. Energy like ATP or the ions for the movement of the elements. So such type of extra energy is needed for the movement. Those type of transport is known as active transport. And next is, they have told about osmosis. So what is osmosis means? Movement of elements from a region of higher concentration to the region of lower concentration is known as osmosis. So why we are talking about all these movements of elements is because it is semi-permeable membrane, the plasma membrane. It is not does not allow the elements to move freely. It has some restrictions. So studies have been made on the movement of elements alone. So the result of those the researches only, these have come. So that is why we are learning about osmosis, we are learning about passive transport, we are learning about active transport and all these things. Also the fluid mosaic model. And the osmosis, if you see, means inside the plasma membrane there is a layer. So inner to the organelles region, the cytoplasm region, where cytoplasm is present, there we can consider it as a lower concentration area where it is not well concentrated. Whereas if you see outside the plasma membrane, the cell wall or the external environment, it is highly concentrated. So what happens? happens when something has to move inside, it is coming from a region of higher concentration where the sodium concentration we can take, where the salt content is very high outside. Whereas if you see in the inside the cell, it is very low. So this is a lower concentration and outside is the higher concentration. So what happens? The movement of elements comes from a region of higher concentration to a region of lower concentration. So this movement is nothing but it is called as osmosis. So these are the things that are coming under plasma membrane. Now I will give you a short recap from the first. So in this video, we are only seeing about the eukaryotic cells. So these eukaryotic cells are present in protists, that is a little more higher fungi, algae, all these things are protists. So protists, plants, animals, even human beings, we also have the eukaryotic cell only. And fungi, in these regions, in these species alone, the eukaryotic cell is present. In well-developed organelles will be present in the eukaryotic cell. So plant cell, if you take, means they have cell wall and it will have very large vacuoles. And animal cell, if you see, means the vacuoles are very small and no cell wall is present, but centrioles are present in animal cell. So this eukaryotic cell, they have a nucleus and around that they have a nuclear membrane. Inside that they have a genetic material. So they are very well arranged. So that is why it is called as well-developed cell organelles are present. So this is all about eukaryotic cell. So now we are moving on to the organelles, different organelles of the eukaryotic cell. So this is the structure of the plasma membrane. So this rod shape is called as integral protein, and this round is the phospholipid head, because it is mainly made up of plasma membrane is mainly made up of proteins and lipids only. So a phospholipid means fat. Phospholipid head is there. Phospholipid tail. Cholesterol is present here and there, and finally glycoproteins are present. So the plasma membrane is mainly made up of phosphoglycerides. And when we are seeing about the plasma membrane of the human erythrocytes, the red blood cells, if you see, means 52% is protein and 40% of lipid is present in the plasma membrane of the human erythrocyte. So there are two types of proteins that are present. One is peripheral protein and the second one is integral protein. So peripheral protein means it will be present on the layer of the protein. Whereas if you see integral protein, it will be buried inside the layer. So these are the two types of proteins. Then we saw about the fluid mosaic model. It was found in 1972 by Singer and Nicholson. So what this theory tells is about the movement of the elements in the plasma membrane. That is, the plasma membrane is selectively permeable membrane or semi-permeable membrane. It will allow only certain substance to go in and out. So studying about this movement of the elements is based on this fluid mosaic model. Then we saw about what is passive and what is active transport. So passive transport means when the energy is needed for the movement of the elements, then it is called as passive transport. And when energy is needed, that is energy like ATP, all those things are needed for movement of the elements, it is called as active transport. When no energy is needed, it is known as passive transport. When energy is needed, it is known as active transport. And then finally we saw about osmosis. Osmosis means movement of elements from a region of higher concentration, the external environment, to the region of lower concentration inside the cell. So that is called as osmosis. So the called as diffusion. From one concentration to another concentration it is traveling. So that is called as diffusion. So from a region of higher concentration it is entering into the region of lower concentration. So it is called as osmosis. The next organelle is about cell wall. We are going to see. So we saw about plasma membrane and now cell wall. So what is cell wall? It is the outer covering of the plasma membrane. So the plasma membrane is there, and over that another covering is there, that is called as cell wall. So what are the different characteristics of this cell wall? It gives shape to the cell. It gives a definite shape to the cell, and it protects the cell from damage if it gets any accidentally, if it gets any wound. So this cell wall, it affects from damage. So it gives flexibility to the cell and cell to cell interaction. If any cell has to come near, they need to interact. The cells need to interact. For that also, cell wall helps for the communication of cell to cell interaction and barrier to undesirable macromolecules. So it protects the molecules that are unwanted to enter, that are trying to enter into the cell. So in the environment there will be lots of unwanted materials. Many big molecules will be present. So they will try to enter into the cell. So this cell wall, they will protect the cell and it will act as a ring. So these are the four main functions or characteristics of cell wall. And the primary cell wall and secondary cell wall are present. So what is primary cell wall? This primary cell wall will be present when the plant is very young. In such a stage, the primary cell wall will be present. So they will help in growing of the cell. So after it gets fully matured, what happens? The primary wall breaks off, and after that the secondary wall is formed. So what happens? At a very young stage, the primary cell wall is present. After it has been matured, what happens? The primary wall breaks off and finally the secondary wall is being formed. And after that, the middle lamella will be present. So what this middle lamella will do is it consists of calcium peptide. This is a component that is present in the middle lamella. So this calcium peptide, it will act as a glue to attach all the components of the cell. So this is the main function of the calcium peptide. It is present in the middle lamella. So what happens is the cell wall and this middle lamella, they will be attached by this component called calcium peptide. So what happens? This will also helps the neighboring cell. The cell is next to the next cell that is present. The cytoplasm of one cell and another cell is connected through this calcium peptide only. So what happens? Here one cell is there, here another cell is there, the cell wall is present and cytoplasm will be present, and center we can find vacuoles. So what happens? The cytoplasm of this cell and the cytoplasm of this cell is connected by some means. So that is called as plasmodesmata. When the cytoplasm of one cell and another cell are connected with the help of calcium peptide, that is called as plasmodesmata. There is a connectivity between one cell and another cell. So that is called as plasmodesmata. So this is all about the cell wall. And next is endomembrane system. So endomembrane system means under this the endoplasmic reticulum, Golgi complex, lysosome, vacuoles. So these are different types of organelles that are present in the cell. So these are, when these organelles together called as endomembrane system. So endomembrane system consists of endoplasmic reticulum, Golgi complex, lysosome and vacuoles. So first we are going to see about endoplasmic reticulum. So this endoplasmic reticulum, it is of two types. One is smooth endoplasmic reticulum and another is hard endoplasmic reticulum. So there are two types of reticulum. So when the reticulum has the presence of proteins in it, it is called as rough endoplasmic reticulum. When the proteins are absent, they are known as smooth endoplasmic reticulum. I will later on explain you with the diagram. Now let me move into the types of endoplasmic reticulum. So this endoplasmic reticulum, if you see, means it is a folding like structure, a network, a continuous layer of foldings will be present in this endoplasmic reticulum. So they are divided into two compartments. One is luminal compartment and another is extraluminal compartment. So this will be present in the external part of the endoplasmic reticulum. Luminal compartment is present in the internal part of the endoplasmic reticulum. So these are the two compartments of endoplasmic reticulum. Now I will give you a short recap from the first. In this part we saw about cell wall. Cell wall is nothing but it is an outer covering of the plasma membrane. So outer to the cell wall, outer to the plasma membrane, the cell wall is present. So the functions of the cell wall are, it has shape, it gives shape, it protects from the damage. If any damage happens, this cell wall helps, and it gives cell to cell, helps in cell to cell interaction. For any cell communication, this cell wall only helps, and it is a barrier to undesirable macromolecules. So around the environment there are a number of undesirable components that are not needed for the cell. For the protection of that, for acting as a barrier for those molecules, this cell wall is present. So these are the four functions of the cell wall. And we saw about primary and secondary cell wall. So primary cell wall will be present at a very young stage, and after maturity the primary cell wall dies, and after that the secondary cell wall is formed, and the middle lamella. So after the cell wall, the middle lamella will be present. It is made up of calcium peptide. This is a chemical compound, and it will be helpful in it acts as a sticky agent to attach all the components that are around the cell. So what happens due to this presence of middle lamella and the cell wall, the cytoplasm of the one cell and cytoplasm of another cell is getting connected like this. So the cell wall is there, cytoplasm is getting connected, and the vacuole is present. So this attachment of cytoplasm of one cell and another is called as plasmodesmata. And then we saw about endomembrane system. So endomembrane system means it consists of endoplasmic reticulum, Golgi complex, lysosome and vacuoles. So organelles together form the endomembrane system. And after that we are going to see about endoplasmic reticulum. So endoplasmic reticulum, it is a foldings of, it consists of many folding, and it is a continuous channel, and it is divided into two types. One is rough endoplasmic reticulum and another is smooth endoplasmic reticulum. So in rough endoplasmic reticulum, small ribosomes will be present. So it is called as rough endoplasmic reticulum, and when ribosomes are absent, it is known as smooth endoplasmic reticulum. So these are the two types of endoplasmic endoplasmic reticulum. This is the structure of endoplasmic reticulum. So you can see the diagram of endoplasmic reticulum. So the structure will be like this. In the center you will have the envelope, nuclear envelope. Center there will be nucleus to guide the cell, and after that around that a nuclear envelope will be there to cover the nucleus, and on the side you can see some foldings. So the dotted foldings are called, you can find small purple dots, so those are nothing but ribosomes. So ribosomes are used for protein synthesis, and in endoplasmic reticulum, when proteins are embedded like this, it is called as rough endoplasmic reticulum. And on the other side, if you see, means there won't be any dots. So plain foldings will be there. So such type of endoplasmic reticulum is known as smooth endoplasmic reticulum, because there is no ribosome in there. So this rough endoplasmic reticulum and smooth endoplasmic reticulum. So these are the two types of endoplasmic reticulum, and this rough endoplasmic reticulum helps in the synthesis of proteins, and smooth endoplasmic reticulum helps in the synthesis of lipids. Lipids are nothing but fat materials. So this is the structure of endoplasmic reticulum. So the next organelle that we are going to see is Golgi apparatus. So in the last class itself, in the last board itself, I have told you about the endo system. So in that endo system, what are the organelles that we have considered? The endoplasmic reticulum, Golgi apparatus, mitochondria and lysosome. So these are the organelles that were coming under the endomembrane system. So in that system we saw about endoplasmic reticulum. So you know what is endoplasmic reticulum. So next is Golgi apparatus. What is Golgi apparatus? So this is the structure of Golgi apparatus. You can say some wavy like structures and some globules moving around. So this is the structure of Golgi apparatus. So this Golgi apparatus, it will be in flat disc shape, and each curving that you can find here is known as cisternae. So each curving, the foldings that you can find in Golgi apparatus is known as cisternae, and the size of the cisternae is 0.5 mu meter to 1 mu meter in diameter. So the diameter of the cisternae will be around 0.5 mu to 1 mu meter in diameter. So each Golgi apparatus has different number of cisternae. So in some there may be 5 foldings, or in some there may be 10 foldings. So the number may vary in Golgi apparatus. So what it does is, this Golgi apparatus, it will transport all the elements. So it is like a postal service or the courier service that we use to send materials from one place to another. It is like a postman. So what it does is, various elements from the cell will be sent here, and through this it will be transported as secretory vesicle. Secretory vesicle means round. Secretory vesicle, this is the incoming transport vesicle. So the element that has to be transported will come here, and after that it will be transported as secretory vesicle. So this is the main idea of Golgi apparatus. The main aim of Golgi apparatus is to pack the materials and transport it. So this is the cis phase and this is the trans phase, the two phases of the Golgi apparatus, and it helps in the formation of glycoprotein and glycolipid. It helps in the formation of glycoprotein and glycolipid, and it like I already told you, it helps in packing the elements and distributing to the other parts of the body. For example, we can take ATP, the energy molecule. So it will be coming from the mitochondria to the Golgi apparatus, and that will be transported as vesicle to the parts that needs energy. So the endoplasmic reticulum, what did I tell you? It produces proteins and lipids. So how can it be transported? So protein is needed all over the cell. So for that, this Golgi apparatus which help to transport the material. So this is the main function of Golgi apparatus. So next is lysosome. So this is another organelle of the cell. So this lysosome, it is nothing but it is like a digestive system. So we eat in the morning. So what happens? The food is getting digested and we are getting hungry again. So the digested food, what happens? It is converted into energy so that we can do our daily activities. So this is the main idea of the digestive system. So similarly, this lysosome, what it does, it will digest the materials that are present in the cell. It is like a digestive system, lysosome. So you can see the structure of lysosome. So there is a two-layer of phospholipid, like the plasma membrane. Here also in lysosome there is a two-lipid layer, phosphobilipid layer, and in the center you can find some proteins and around it some enzymes. Those enzymes are nothing but the digestive enzymes. To digest the food, these enzymes are present. So this is the structure of the lysosome. So it is a membrane bound vesicle. So why it is called as membrane bound vesicle? Because it is round in shape. So it is called as vesicle, and it has a membrane. It has two layers of lipid. So it is called as a membrane bound vesicle. And like I already told you, it will produce some digestive enzymes. For digestion, it produces digestive enzymes, and it is known as the digestive bag. So lysosome is known as the digestive bag, and the function is digesting microorganisms. So the function of the lysosome is to digest all the small microorganisms. So this is the main function of lysosome. So till here I will stop. You have some organelles also in your lesson. We will continue it in the next session. So till here I will stop. So in this class we saw about what is eukaryotic cell. So eukaryotic cell means it is nothing but it is a well developed cell organelle, and the structure of what are the elements that are present in the organelles that are present in eukaryotic cell. So we saw about plasma membrane, we saw about the movement of the plasma membrane, why it is called as the semi-permeable membrane, about fluid mosaic model, transport, active transport and passive transport, osmosis. So all these things we saw, and we saw about cell wall, the endomembrane system, in that endoplasmic reticulum, Golgi apparatus and lysosome. So these are the topics we have covered. There are other organelles that are also present in the cell. I will tell you later on. So till here we will stop. PDF will be given, the notes PDF as usual. So copy all the answers including the detailed answers in your note and submit it in the given day. Thank you, children.